Hello and welcome to the Point Blank Show. My name is Abhishek. Alzheimer's affects over 30 million people worldwide by some estimates, with the risk doubling every 5 years after the age of 65. It is a slow and inevitable decline, and it's quite devastating for patients as well as their families, their loved ones. Despite being one of the biggest public health challenges, there is no cure. Yet, in 2006 and 2012, two quote-unquote groundbreaking studies claimed that they had cracked the problem, but their assessment was based on falsified data, as Charles Piller, investigative journalist for Science, went on to unearth. His book, doctored Fraud, Arrogance and Tragedy in the Quest to Cure Alzheimer's, exposes this shocking deception in the field. It's an important piece of work, and Charles, thank you so much for taking the time to talk to me.
Well, thank you for having me. I'm glad to be here.
Thank you. Charles, you are an investigative journalist. That is a field in itself, and one that I got introduced to after having watched that movie, All the President's Men. Obviously, things must have changed since then, and it's a dramatic representation, all right. But in this story that you went on to discover, you have science. It's a difficult one for a layperson to understand. You have to make it palatable to the audience who have no background in science, and yet it's an important subject for people to know. So you have molecules, amyloids, I can't pronounce some of the stuff that you've written from the science standpoint, and proteins, and at the same time you have the human element to it. Let's start with that. I mean, what was your experience of meeting patients, their loved ones? What do they share with you? What do their, guardians and those around, when you spoke to them, what does that come about? Because not every one of our listeners would have a direct link with a patient in their house or extended family that has this scourge, but it's a big one.
Sure. Well, it's very much a human story, and the way that I tried to structure my book is around the players in this story: patients, their loved ones, the scientists, both those who have been accused of misconduct and those who have exposed it, and the path that each of those players follow, partly because, as you say, there's a lot of technical information to understand the science behind Alzheimer's disease, but there's also a very human story that anyone who has a loved one who's been unfortunate to be afflicted with this terrible neurological ailment understands that this can be devastating for families, and also a gigantic human challenge for all of us because of the growth of the problem and the extent to which it is being affected by our improving lifespans. As medical developments, medical science has increased lifespans all over the world because of these important breakthroughs in vaccines and in treatments for cancer and heart disease and other ailments, people are growing older, and as they grow older, they become more susceptible to various forms of dementia, including Alzheimer's. And so critical to my story was to introduce a patient who could represent the human experience of not just having Alzheimer's and what that's like, but also what is it like to be the subject of an experiment, of a drug that was designed and meant to help treat or even cure the disease. And it's that human story. And unfortunately, the betrayal of that patient who ended up being involved with a drug that was shown to have been based on a lot of improper, some would say fraudulent data, that kind of set the stage for a bigger discussion of what ails the Alzheimer's research institutions.
And these patients too, Charles, and you talked about this individual in particular, they seem to be on trial at all times, don't they? Meaning, you write this a little insight, you give us into the book where you talk about that person cracking a joke and then looking around for a reaction because they are always being judged. Is this guy serious? Is he? And he wants to show that, hey, listen, I have not lost my mind completely and I can crack a joke here and there. So come on, guys, loosen up a bit, is what he must think, I wonder.
I think so. Obviously, one has to read between the lines when talking to a person who has developing Alzheimer's because, first of all, we all need to remember that having empathy is critical, not just to helping Alzheimer's patients, but for me, the reporting process. As a reporter on this topic, I have to keep reminding myself that these folks, they're living their experience in ways that I can only imagine. And I have to remind myself to not just be sympathetic, but to take them seriously where they come from. In this case, you're talking about an individual in my book, a guy by the name of Stephen Price, who is living with Alzheimer's. And yet also you see that at every opportunity he's trying to keep hold of the humanness that makes each of us remember that life's worth living even if we have diminished capacity. And one example of that is that he used to be a construction guy back when he was working. He's now retired, of course, and because of his advanced disease wouldn't be able to do that work anymore. But he still enjoyed going into his shop and moving around tools and thinking about his old experiences. And that's the sort of simple respect that I think all of the people who are living with Alzheimer's deserve. It's the kind of respect that remembers that they have a real life that they're living, and we need to just address them with the kind of way that we would like to be treated and respected in our own situations as well.
Right. And one thing that baffled me is that for all the advancement in medical science, when it comes to curing or at least preventing or helping a patient through, whether if he has a heart disease or a stroke or cancer, AIDS, there has been a lot of development, even cardiovascular diseases, but not so much in Alzheimer's. Would that be right to say? And what's the reason? What's that big challenge that the entire scientific community is trying to grapple with, and we don't seem to have a cure? And it's a decline which you know is coming, which is the dreaded part, isn't it? That you can't do anything about it. Things will get worse.
Yeah, I would say that's an excellent question. And here, I think the answer is a little bit more complex, which is that, first of all, what we've seen over the last few decades is that the rate of death from heart disease, from stroke, from cancer, and other very important diseases has been declining. That's because there have been terrific advances by medical science. That has not been the case with Alzheimer's disease. As the population of older people grows, when lifespans grow, we have seen increased death rates with people who are associated with their Alzheimer's. And there's a couple reasons for it. One is that, let's face it, this is a tremendously challenging and difficult medical problem to solve. The human brain is such a complex organ. The biochemical, biomedical challenges for studying brain science and brain disease are enormous. And I don't mean in any way to minimize just the basic scientific and biomedical hurdles that scientists and doctors are facing. So that's number one. The other is that there has been scientific misconduct related to basic and clinical research in all biomedical fields for really since time immemorial. This is part of the human condition. Most scientists, the vast majority, in my opinion, are honest. They are deeply determined to do something beneficial for human beings. They are working with integrity and honor, but always, like in any walk of life, there are always a small percentage of people who cut corners, who might be falsifying information for personal gain, or who might have even decided that their ideas are true and correct, even if unproven, and they'll do anything to promote those ideas, even if it means falsifying information. And what we've seen is that even though there has been some of this behavior in cancer research and in heart disease research, there has also been enormous breakthroughs in those areas that have so much benefited patients. But not so in Alzheimer's disease. There have been many examples of falsification of data and of improper activities in research and in research integrity questions and in real fraud that have resulted in skewed thinking in the field, have resulted in setbacks and wasted time and wasted funding, which is precious in doing research and particularly in clinical experiments. And what the result has been, in part, of this combination of the difficulty of solving this problem of Alzheimer's and the falsification or misrepresentation of scientific information has been that there has been no remedy developed that either arrests or reverses the terrible cognitive symptoms of Alzheimer's disease. And the products that have been developed that are on the market now to address those symptoms have been, again, this is a much longer conversation which I'm happy to discuss, but I think we can say that even their strongest advocates would acknowledge that their benefits have been at best extremely modest and still result in the steady decline of cognitive abilities in Alzheimer's patients who are taking those drugs.
And I wonder, even measuring whether a drug is working will need some time, won't it? Like a heart attack, or rather, when you have a stent, you create a valve in a bypass surgery and you know that you will live for the next 25, 30 years because you fixed that little bit. You won't be panting when you are running up a flight of stairs. How do you measure the effectiveness of a drug over time? It must be hard, right? It's like pain. There is no unit of measure for pain. You have to, on a scale of 1 to 10, they ask people to put a figure next to it when they are being treated for pain medication, for instance. Is Alzheimer's similar in that respect, that it's challenging to know what's working, what's not?
Yeah, it's a terrific question because it's central to the evaluation of whether these drugs are being effective. And so the way in which they're judged is based on a certain developed testing that involves questions and drawing pictures and memory problems and things like that. Now, these are not trivial tests. Many of them are validated and quite good, but there are problems in using them that make it chancy to know with certainty how effective drugs are if their benefits or their ostensible benefits are subtle, which is the case with the drugs that are on the market now for Alzheimer's disease. And these challenges are one that they're subjectively evaluated. So the expectations and the perceptions of the test administrator, often physicians or other medical professionals, and also of family members who are involved in this and of the patients themselves, can affect the outcomes. And so there can be something called response bias. So that's pretty important. The other element of it is that Alzheimer's patients, as anyone who is a family member of a person who's living with Alzheimer's knows, they have good days and bad days. And so the time of the day, the particular day that a test is administered can affect the outcome. And so one has to take this testing with a bit of a grain of salt, because we know that although it tells you something about the patient, it doesn't tell you everything.
And at what stage, your time is at a premium, Charles. You've dug yourself in for a number of years to first write the story in Science, which was titled Blots on a Field, with the rubric that said a neuroscience image sleuth finds signs of fabrication in scores of Alzheimer's articles threatening a reigning theory of the disease, unquote. I'm reading it off your site. And this itself came about after a lot of research, and you, after having verified it through independent experts, what the whistleblower, which I will, would love to hear from you about him, and the kind of work that went in. At what stage did you realize that this is not just an isolated case of bad science, but something bigger, and this needs to be told? And secondly, was it surprising that why isn't everyone talking about it?
Yeah, that's an important way to frame it. Thank you for the question. Let me, if I may, put it into historical context, because I think it's important to really understand the evolution of how people thought about this disease. So Alzheimer's disease was first described in 1906 by the namesake German scientist who observed he was both a clinician and a pathologist. So a pathologist is someone who examines tissues, and often these are after the death of a patient. And so this Dr. Alzheimer, what he did is he examined the brain tissue of a patient who had died after suffering from extremely terrible dementia symptoms. And what he found in her brain was a combination of two proteins. And one is these deposits in the brain of something that's often been referred to as sticky plaques. And these are amyloid proteins. And the other form of protein found inside brain cells is something called tau proteins. These are described as tangles after their kind of tangled string shape inside the neurons of the brain. And he said the combination of plaques, tangles and dementia, that's Alzheimer's disease. That's a disease. He said it wasn't him who named it after himself, but it was eventually named after him for first describing it. And so this was very interesting, but not an important source of study for the neuroscientists of that era. In fact, not for many decades did anyone pay super close attention to delving deeply into Alzheimer's disease. And the reason is simple demographics. Until breakthroughs in vaccines and antibiotics and cancer drugs and heart drugs started to greatly extend lifespans, we didn't have a critical mass of Alzheimer's patients, people who had reached the old age that the vast majority of people who are unfortunate to come down with Alzheimer's disease, the old ages that occurs at. And so consequently, in the 70s and 80s, when lifespans were increasing and many more Alzheimer's patients were seeing physicians, there was a much bigger emphasis on research. And so in the early 90s, an idea was developed, this is sort of in the 80s and then finally coming to fruition in the early 90s. An idea was developed that came to be known as the amyloid hypothesis of Alzheimer's disease. And it was really pretty simple to understand. It held that these sticky amyloid plaques, deposits of these proteins in the brain, and also eventually they decided other forms of amyloid protein that were separate from the sticky plaques but the same class of protein, kicked off a cascade of biochemical effects within the brain that eventually caused cell death and led to the dementia symptoms of Alzheimer's disease. And this was a very sensible and well-reasoned way of looking at the disease because there was such strong evidence to suggest that people with Alzheimer's disease did have the presence of these proteins. It was a logic that really found its genesis in Dr. Alzheimer in 1906 in his initial observations, and it became the dominant way of looking at the disease. But during the 90s and early 2000s, a number of drugs and even a vaccine were developed to attack these amyloid proteins and remove them from the brain. And these remedies, these drugs and this vaccine were extremely effective in doing that, in removing these amyloid proteins. But they were very ineffective in helping patients with Alzheimer's disease either arrest their symptoms or become better. And consequently, a lot of discouragement had entered the field. They thought, many scientists were beginning to say, maybe we're on the wrong track, maybe this is a problem of a complexity that we haven't quite figured out yet. And there was doubts being made about this amyloid hypothesis and where it was leading the field. So then, now I'm finally circling back to answer your question directly. So then in 2006, there was an important experiment done at the University of Minnesota in the United States. And this experiment worked with genetically engineered mice that were produced to make copious volumes of amyloid proteins in their brains. And they extracted some of these proteins and purified one particular kind called amyloid beta star 56, the so-called star protein. And they injected this into rats and found that the rats displayed symptoms of memory loss that were akin to the memory loss that people experience with Alzheimer's disease. So even though this was an animal experiment, people realized, or at least decided, that they had found the first substance ever that was shown to have a cause and effect kind of relationship between a substance and the symptoms of memory loss. And they thought, oh, this really demonstrates that we are, after all, on the right track with amyloid proteins. Maybe we haven't found the right formula. Maybe we haven't targeted the specific type of protein that is most beneficial to look at in our drug development, but we are really on the right track here. And it was a famous experiment that became one of the most cited experiments in Alzheimer's research for a period of many years following that, and was not just cited in the literature, but cited by the leading figures in Alzheimer's disease, the chief proponents of the amyloid hypothesis, over and over and over, despite the fact that other labs were having terrible difficulties in reproducing the results, or even being able to effectively identify and refine this star protein, amyloid beta star 56, that was identified at the University of Minnesota. So this is where I finally start to enter the picture. with my own reporting, I became connected with an important scientist in this story, a guy by the name of Matthew Schrag, who is a neurologist who treats patients at Vanderbilt University in Nashville in the United States. And he is also a neuroscientist who runs a lab studying Alzheimer's disease. And so Schrag and I had been talking for quite a while about a particular set of experiments from a company called Cassava Sciences that we can talk about if you'd like, which is another part of the story that I tell in my book. But during the course of his examining the experiments, which were suspect and ultimately shown to have been based at least largely on doctored images for the drug that Cassava was developing, he ran across experiments done by a scientist at the University of Minnesota who has ended up being one of the principal people behind this important 2006 study on the starfish protein. And this study, I might add, was published in the scientific journal Nature, which is one of the preeminent authorities on science in the world. And so that was part of why it gained such fame and became so important in the scientific community. So what Schrag found was that a number of studies by this scientist at the University of Minnesota and his mentor, the two of them, that were apparently had suspect images, doctored images. When I say doctored images, what I mean is that these scientific images were these scientific images were changed using tools like Photoshop or other digital tools that can erase certain elements of an image or move parts of it around or duplicate certain elements of an image or reuse them in improper context to establish that a hypothesis was proved in an experiment when actually the real data refuted that or didn't support the hypothesis. So this is pretty important. And when he realized that one of the studies that was suspect because of the apparently doctored images within the study was this a very important study in Nature, it was kind of a revelation. And I remember the moment that he and I were talking about this for the first time, and we both lapsed into a bit of stunned silence during our conversation because we realized that this seminal experiment that had been quite influential in the field, if it was based on doctored data and if it was in fact a false experiment, it could have real ramifications in the understanding of the importance of the so-called amyloid hypothesis that was quite dominant still in the field. And even though that hypothesis had been supported by other experiments and there was disagreements within the field about its importance and its centrality to thinking about Alzheimer's disease, this experiment was so important that it certainly, we knew, if his findings were true, would raise big questions in the field. And that sort of was the outset of my several-year journey myself through looking more broadly at Alzheimer's research and the meaningfulness of possibly doctored findings in the field.
Right, and that's where you found that it's worth your time and that of your editors to go and chase this story. What I find common sense as a layperson was not so common in the community of these experts is what my inference is. And why I say that is, A, if you can't replicate a finding, even in software, if there is a bug and you report a bug, you actually share steps that the developer can reproduce that particular bug so that he or she can fix it. In this case, two things happened. As you mentioned, that despite the study being so popular and many great scientists quoting it over and over again, how could they get away with all of that when there is a bunch of people on the other end who couldn't replicate it? So what were the vested interests that allowed this to spread so much so that were people squeamish not to pass a rebuttal or tell these journals that, hey, you guys are being stupid, with due respect to you, because this simply doesn't work? This is how I would see it as a third person, because I am the guy, believe it or not, Charles, the other day I was having an argument with a friend of mine saying if a journal is peer reviewed and if it's coming from the likes of natures of the world, you have to believe science is real. And he got into a tiff with me. No, you don't need 7 hours of sleep. All you need is rest. And I said, what is your source? And I got into an argument, or rather a reasoning with him, that if it's in a peer-reviewed journal, it has to be true. And then I read doctored. And I have to give him a call now and say that, hey, you can't, I mean, despite it having those sources, you never know until somebody like you walks in and, puts it under a scanner. How did somebody miss such a big thing? Sorry, my question was too long there.
Yeah, it's okay. There's a lot of, let me unpack that a little bit and reflect on a few of the issues that you raised. So, first of all, we have to remember that science is a human endeavor. And even if there's no misconduct going on in a particular experiment, there can be errors made because each of us is human and everyone makes errors. Everyone may have false ideas that are maybe changing our thinking about something that would benefit from other review or examination. So these are not always matters of misconduct, but nonetheless, the peer review process that you're describing, this is where experts are asked to read articles before they're published and to comment on them, and it's usually a rigorous process. The problem is that when it comes to misconduct or image manipulation in science, peer reviewers are not asked to, nor are they qualified in most cases, to examine that aspect of an experiment. And consequently, peer review very rarely would catch those sorts of problems. Now, that doesn't absolve the journal from their own responsibility to be the gatekeeper of correct scientific knowledge. And what we found, and what I've found, and it's detailed in my book, is that journals are playing catch up on this. They are, unfortunately, have not historically been very vigilant in examining scientific papers for possible fraud or misconduct associated with scientific images. And they're getting better, especially the most prestigious journals, including Nature, which we mentioned before. The journal that I work for is Science, and Science and Nature are both regarded as important, very important journals in the scientific world. Both of them, among lots of other journals, have in the last few years started to implement very much more rigorous controls to try to find these things. Now, still, stuff can slip through because it's hard. It's difficult sometimes to detect deliberate misconduct. But that said, as a general rule, a lot more work needs to be done about this. So that's problem one.
Right.
And also, I think we should remember that there are some, what I would call, perverse incentives within the scientific community and the academic community. One is that even when people might suspect or find incorrect results or not replicable results in an experiment, it can be very difficult to get that published. And particularly if that is questioning an experiment from a prestigious lab or a prestigious scientist. Because people tend to revere and respect and believe important scientists, and questioning their work can be difficult, but also can be perilous, especially for scientists who are still trying to make their name in their field, for young scientists or people who are still beginning to develop their career, who might be skeptical of an idea, but they can't really challenge it or question it. One, because it's hard to get it published, sometimes impossible. And second, because it can be damaging to their career, and as a result, they're reluctant to take that sort of action. So that's in part why a guy like Matthew Schrag at Vanderbilt University, he's a courageous guy, because he was a junior scientist who did not have tenure in his institution when this all came about. We went through a long process of trying to determine, he did, I should say, with my involvement, went through a long process of trying to determine whether he should be named in my articles in Science and in the book, because he was someone who was really risking the possibility of the wrath of journals and funders and important scientists in his field by exposing apparent misconduct. But there are people like that who are willing to do it, so that's very important. But I think, just to summarize, what we have is these perverse incentives, which mean that a lot of times people are asked to, the publish or perish incentive, which means that quantity over quality is sometimes what you get in order to advance in your career. The second is that when you can't replicate a finding, it can be difficult to publish that in journals. And third is the fear of challenging important scientists in the field. All of these kind of, I use the word conspire together, but in a loose sense, it's not an actual conspiracy, but they are factors that make it difficult to uncover and expose and change bad thinking in the field. And let me just, if I may, give you an example of what happened when I originally wrote about this famous 2006 study in Nature in the pages of Science magazine. This is long before my book was published. This is back in 2022. There was an immediate, gigantic reaction within the field of Alzheimer's research. And the reaction was really two kinds of reaction. One was people who said, we knew all along this was suspect research. No one said so over these many years. This, to me, was rank hypocrisy, because many of the people saying this were the very people who were citing this study over and over and over in their own scientific papers as support for their ideas, not just about their own scientific research developments, but for the validity of the amyloid hypothesis itself. And so, to me, those were not credible comments. The other side of it were people who said, finally, we should take a good hard look in the mirror and examine whether some of our assumptions about the amyloid hypothesis and other aspects of our study should be deserving of a very close second look. So to me, it was very interesting how the field responded, and it reflected how difficult it was and how difficult it is to change entrenched thinking in the field, even when evidence contrary to that set of ideas may come to light.
And you said that not all of them are crooks. In fact, this must be a minority that brings a bad name in any industry, science no less. Even those who are bad actors, I would imagine, started off with good intentions. I mean, nobody gets into the field of science to cheat, say, or to falsify or create an empire or have a resume which is based on false. That's not how a 10-year-old or a 15-year-old started thinking about wanting to be one. So where is it that, I remember there's a movie called LA Confidential. This suddenly comes to mind, where there is a young cop asking his senior officer who's taking a lot of bribes and says, why did you become a cop? And he pauses for a second, and Kevin Spacey, he says, I don't remember. And he gets that epiphany that I need to fix this thing. At least for this moment, let me make an exception and chase the bad guys. I'll go back to my old methods later. But he has that little... So I'm assuming that nobody gets into this to do these kinds of things. You talked about image doctoring. In that context, what exactly happens? Is it that, let me take a sip. Let me do a little doctoring here and it's helping my hypothesis. Let me go one step ahead. Could you take me through the process of what that image doctoring is and how does it work? I may be speaking out of thin air here, but help me out.
No, well, actually, I think you're onto something there. So I've thought about this a lot, as you might imagine. And because it's sort of central to what is the human element of this? How does it evolve? And in order to understand that, I think we just have to remember that human motivations can be complicated. And I am not a psychologist, and I can't read minds, but from my observations over these years of examining case after case after case of this, what I surmise is the following. I think often it starts with this idea of quote-unquote beautifying an image. So scientific images are important because people should remember that these images, they are the data of experiments. It's not just an illustration of something. The image itself is the data. It's the proof of an experiment in many cases. Consequently, if you alter the image, you're altering the data. But there are gradations of this, as you suggest. So, for example, you might want to improve, I know there's an expression in the United States, curb appeal, when you're selling a car. So you want to polish it and make it look as good as possible. You're not changing the engine of the car, but you're prettying it up so that more buyers would be interested. I think there's an element of that goes on within scientific publishing, because journal editors are like all of us. Not only do we want truth, we want beauty. And so if an image looks good, and what I mean when I say looks good, I mean from a scientific perspective, if it has the sort of inherent beauty and order that one would imagine from experts in the field, then it enhances its ability to be published. So doing small modifications along those lines, they're scientifically improper, but they're usually, people don't take it that seriously. But what often happens, I think, is that someone might get a weak result in an experiment that's only very weakly demonstrated in their results. And they feel maybe that they could enhance the way it looks in an image by making a certain portion bolder or a certain portion less bold by perhaps altering just a small portion of it in order to enhance the weak result with the justification of saying, well, I did get my result. It's maybe not as strong as I expected, but I think it's going to be important, and I think this will help get this paper published, and that will help me get more funding in order to be able to do new iterations of this experiment that can finally prove it out. And that's, of course, improper, but because it can be subtle, it's often missed by journal editors. Then you maybe get to the point where someone becomes so convinced of their ideas that even if they don't get a result that they're after, they just reproduce it by improperly changing an image to represent that result. And of course you have the cynics and the crooks who make images a different whole cloth because they simply are doing it for personal advancement or possibly for profit, as can be the case in some drug-related studies. But I wonder if it would be useful to describe specifically what kinds of images I'm talking about, to give people a mental image of what we're looking at specifically. So in Alzheimer's research, there are two types of images that come up over and over and over again that seem to be central to a lot of the science that's done in the basic experiments in the field. One is images of brain tissue, and these are images that show the individual cells sometimes they show the effects of drugs or treatments on cells, sometimes they show the way in which cells affect, the way in which the different portions of the brain appear under different circumstances for an Alzheimer's patient, and these can be changed by accentuating certain elements of the image, by making them bolder or erasing certain parts that may contradict an experimental hypothesis. And they can also be reproduced in the wrong circumstances. So for example, they can be used out of context, reproduced in a paper 2 years or 3 years later that is a different hypothesis, but this old image might tend to support that hypothesis. So that's completely improper. All of these are techniques that are improper that are very often seen when image doctoring is involved. The second kind of image that is very prevalent is one that displays different kinds and amounts of proteins. So these are like the amyloid proteins that we talked about earlier, and it's a particular process that represents the proteins photographically on a scale that by molecular weight. So this is the relative weight of the proteins, and they appear as a kind of series of stacked bands, kind of almost like a ladder-like series. These are somewhat irregularly shaped, ovalish bands. And what scientists who have been doctoring these sorts of images, they're called Western blots, often have done is to remove certain bands with a program like Photoshop and place them in other locations to reflect the ideas in an experiment that maybe, well, we found a certain kind of protein that's important to the thesis of our ideas, and yet they didn't see it in the data, so they just moved a band up to where that should be to reflect the experimental hypothesis. Or they might have reduced the intensity of certain of these bands photographically in such a way that it makes it look like there's less of that particular protein in a sample. Again, maybe to support an experimental hypothesis. And this is the sort of image doctoring that you see over and over and over and over again in Alzheimer's experiments that makes you realize that, there's just a substantial amount of this going on. Again, in my view, and I think in the view of people who have studied this closely, a small minority of the overall experimentation in the field, but enough to skew certain thinking, to accentuate certain ideas in a way that's improper, to shift funding to ideas that are not fruitful but might benefit certain scientists, and to raise questions about the veracity of certain directions in the field that I think are troubling.
And this has wider implications. Every subsequent study will cite that previous study which is fraudulent or falsified data, and the direction that it's going in the wrong direction altogether. So all the funding that goes is based on a false premise, and there isn't enough incentive for scientists to look for some other way of solving the same problem, because everybody is focusing on some sort of hypothesis, which I think you talk a little bit about the mafia, around that particular way of solving a problem, which is amyloid mafia. Is that
Yes.
if my memory serves me right? Yeah. And would it also be true that there is a lot of competition among these journals? Everybody wants to have these groundbreaking discoveries published. There was in fact an article in The Economist not too long ago where there was one in 2013 apparently there was an experiment that was conducted by one Dr. John Bohannon. He's from Helsinki, and he deliberately doctored stuff while he sent his hypothesis or paper to different publications, and he says, and I quote, he says it was laughably bad methodology, and it had a shocking conclusion that the molecule is a quote promising new drug unquote without any clinical trials, and some 69% of some 121 agencies or papers that were blacklisted by some other agency through its investigative work, they agreed to publish it. And some 38% of the whitelisted ones, which was some 167, the pool, 38% of that said okay. So are the standards getting lax? And I wonder what the regulators are doing, which is the FDA.
Yeah, there's a lot of questions and concerns about the scientific publishing industry. So I think many journals are rigorous, are careful, and although they're a human endeavor and mistakes are made and problem papers are published, they are often extremely reliable, generally extremely reliable, and people can have a lot of faith, even if, science is an iterative process. Even good science can be disproved later. And it doesn't mean that there's any misconduct or fraud involved. This is normal for science. It's a human endeavor, and it's complicated, and people find things that appear to be right that later are disproved. And that's fine. That's part of the scientific process. But it is true that the scientific publishing industry also has a lot of journals that are unreliable, some of which are using improper or even fraudulent methods for profit. And there's a kind of phenomenon called paper mills that are even generating fake papers and selling authorships to papers and doing things that I think are widely viewed as really undermining public confidence in scientific publishing. And it's a huge problem that the scientific publishing industry and regulators and scientific authorities in different countries haven't gotten really into hand yet. I think the bigger question here is that the institutional authorities of science have a lot of work to do to improve their performance in these areas. So my book talks a lot about the institutional authorities of science, and by that I mean the journals, the funders, like in the United States, the National Institutes of Health, which is the largest funder of biomedical research in the world, the regulators, again in the United States, the Food and Drug Administration, but each country has its own drug regulators who must pass muster over the safety and effectiveness of new drugs. And finally, the universities, who are important also gatekeepers of human knowledge. And so these four institutional authorities of science all have been complacent, I think, and lax in their willingness and rigor in both finding and preventing and examining potential misconduct in a variety of fields. And of course, I'm obviously much more familiar with Alzheimer's than others. But what we've seen time and time again is that problems are not examined appropriately. The people who are involved in these problems are often let off without a good examination or care by the institutional authorities in the problems associated with their work. There's a lot of procedural problems and also funding problems for the need to do rigorous reviews of suspect research. And so this exacerbates the problem. It also undermines public confidence. And what I'd like to say briefly is that I don't know how your listeners have been thinking about these issues of what's going on in the United States right now associated with the Trump administration's actions on scientific institutions, but there's been an enormous effort to defund a lot of research in the United States from the National Institutes of Health and to essentially slash and burn, I would use that expression, to address some of the issues and challenges in Alzheimer's disease and other fields of biomedical research, I want to say that even though I have written a book that's very critical of the performance of these institutions and basically has taken the position that we as consumers of medicines and as people who care about biomedical science, as people who love science like myself, who feel that it's incredibly important in advancing human development and incredibly important for our health and well-being, we need to be demanding that these institutions get better at what they do, to be able to catch misconduct more effectively and prevent it more effectively and be more vigilant. But I strongly disagree with the approach taken in Washington, D.C. Even though there are real problems to solve, taking the approach of slashing and burning these agencies and removing the people who have the institutional memory and wisdom that has often served us well is exactly the wrong way about going about it. And so where I find common ground with people who have been critical of my book and who have been defensive about their approach to science, where I find common ground with them is that we all agree that there needs to be a much more thoughtful way of reforming these agencies to the degree that they require reform to do their jobs better and more effectively.
While all of this is going on, Charles, is there hope at the end of it all? Despite the doctored images, certain bad actors, FDA sometimes sleeping on the wheel, you also have conflicts of interest that you bring out where folks on the FDA have vested interest because 2 years down the line they may be recruited by the company that they are currently regulating. Much like the rating agencies didn't do a great job during the subprime crisis. I wonder if there is an analogy there. And with all of this, you have the human angle that you started out with. People might, die a miserable death if this is not addressed. Is there hope or are we doomed?
Well, first of all, let me say that science is vast and there is always a diversity of research going on, even if there is a predominant way of thinking about something. So in Alzheimer's disease, I think we're beginning to see more and more research that is taking a new approach different from the so-called dominant amyloid hypothesis, even though that hypothesis is still the dominant way of looking at Alzheimer's disease. And let me just make it clear before I talk about these alternative methods that I am not saying that the amyloid hypothesis or the role of amyloid proteins is zero in Alzheimer's disease. I'm saying that it's a complex disease and that there may be a number of factors that influence it. Not that amyloid proteins have nothing to do with Alzheimer's disease, just that they don't have everything to do with it. And so there are alternative thoughts about what might be influential in treating Alzheimer's disease. And so one of these are the GLP inhibitors that we've been hearing about so much, these drugs that have been so influential in recent years for obesity, diabetes, and now are being used or proposed for a wide range of other ailments, these kind of so-called miracle drugs like Wegovy and others that have been marketed globally for these ailments. There are experiments going on right now about their possible benefits in early stage Alzheimer's disease. And these clinical trials will come to fruition really as early next year. And so we may very soon know whether the early, potentially promising benefits of these drugs could be also applied to dementia. So that's one thing that I'm looking at quite carefully. The other is really fascinating science behind the idea that infections, latent infections in the brain, may be also a factor in dementia and Alzheimer's disease. And there are studies going on that, for example, that herpes virus, which is a very common virus that causes cold sores, and then those resolve and people kind of figure that they're done with it. These can actually be latent in human organs, including the brain, for many, many years. And there's thinking that by aggressively treating those, there can be another avenue towards treating Alzheimer's disease, that they may be a source of it. And there's good basic science to suggest this might be true. There's clinical trials going on with that approach that within 2-3 years we may have some solid answers on. So that's one I'm pretty interested in. Additionally, I want to say that something that I always want to remind myself of is that we should never let the pursuit of cures, be something that prevents us from concentrating on prevention. And while I do not believe that there's any nutritional supplement or brain game or lifestyle change that absolutely prevents Alzheimer's, of course there isn't anything that I know about that would do that. What I do know is that there are risk factors that we all can have some control over. High blood pressure, high cholesterol, eating habits that are unhealthful, sedentary lifestyles. These are all things that we can have an influence over in our lives. In other words, if we can live our best lives, our most healthful lives, and being conscientious about these risk factors, we can, some of us, many of us, forestall the onset of Alzheimer's disease or have the disease be less severe in its early years than it would otherwise be by simply living our best lives that benefit us in many other ways. So people should remember that we have agency in our lives. We have agency even in the terrible ailment of Alzheimer's disease.
Well, on that very hopeful note, Charles, I think I'd like to thank you for being part of this conversation, for making time, and congratulations yet again on Doctored. One final one, what kind of sacrifices did you have to make when you wrote this? I read one line in the book where I'm not sure if your wife was too happy about the fact that you were already putting some finishing touch to an other investigative piece during your holidays, and then you took this assignment up. And you smile about it now, but this takes a whole lot of work. How did you... Did you ever think this is too much for me? What kind of sacrifices do you have to make to break a story and write a book like this?
Well, yes, an enormous amount of work, and also, some degree of personal risk, because when you write controversial articles in a controversial book that challenges conventional wisdom in places, there's a lot of people who, become angry about it. And I've written articles that have had big and deleterious effects on people's careers, I think quite justifiably. It can result in threats and what have you. But the main thing for me is that I love doing this work because I love contributing to something important to human knowledge and to having a beneficial effect in helping steer thinking towards a kind of productive way of looking at the world that can ultimately help develop remedies and thoughtful ways of looking at science that can benefit people. So I consider it an honor, and yes, it's hard work, but I'm privileged to be able to do it.
Lovely. Thank you so much, Charles, for letting me in on that part of your life as well. Thanks very much for your time on this podcast.
Okay, thank you for having me. I appreciate it.